Chapter 6 focuses on the evolution of the brain and behavior. Segment 2 begins with Box 6.1, which talks about why we should study particular species. So factors considered by scientists in choosing species to study include outstanding features, convenience, comparison, preservation, economic importance, and treatment of disease. Complicated lives require complicated brains, so strategies that different species use to obtain food are correlated with brain size and structures. Finding novel ways of getting food is related to the size of the forebrain in different orders of birds. The inferior colliculus in the midbrain of bats is larger when they depend on hearing, and bats that depend on sight have a larger superior colliculus. Birds and families that store food for later use have a larger hippocampus. HVC is an important structure for song-producing birds, and a general rule is that the relative size of the brain region is a rough guide to the importance of the function of that region for adaptations of the species. Simpler invertebrate nervous systems provide models of neural function. So invertebrates have much simpler nervous systems with only hundreds or thousands of neurons, opposed to the human brain has 86 billion. All vertebrate brains share the same basic structures. The main structures are the same in all animals. All mammals. Relative size, proportions, and anatomical locations of the brain regions have been subject to evolutionary modification as species have adapted to their unique ecological niches. Each main structure in human brains has a counterpart in the rat brain. Differences between brains of humans and other mammals are mainly quantitative. All vertebrates have nervous systems that share certain main features but differ in others. So the development from a hollow dorsal neurotube, bilateral symmetry, segmentation, hierarchical control, separate systems, and localization of function are common features of all vertebrate nervous systems. The evolution of vertebrate brains reflects changes in behavior. So present-day animals and fossils reveal the evolution of the brain. So two methods of analysis are the use of endocasts, which use the cranial activity of a fossil skull to make a cast of the brain that once occupied the space, and the second method is by studying present-day animals, choosing species that show various degrees of similarity to or difference from ancestral species.